Hello and welcome to the Healthcord webinar series on stem cells. In this webinar, I'll be discussing three types of stem cells that are being explored by researchers, the origin of these stem cells, and how their source impacts the therapeutic capabilities of stem cells. To learn more about stem cells, we have to start with human embryonic development. Life begins as a single fertilized egg. Following fertilization, this single egg undergoes many major changes to produce a baby who has more than 200 different types of cells in the body. All of the developmental stages highlighted here happen in the first week of pregnancy. The fertilized egg goes through several cell divisions to create the first structure of early development, known as the blastocyst. Now, if we focus on the stages before the formation of the blastocyst, these cells in the early embryo are identical. They are undifferentiated, which means that they are not programmed to act or behave like any of the specialized cells in the body. They are also totipotent, meaning that these cells can give rise to any type of cell in the body or even go on to produce an entire embryo. Let's look at an example. Identical twins are a great example that highlights the capabilities of these early stage cells. Monozygotic identical twins are a case where a single fertilized egg splits off during early stages of development and give rise to two separate identical babies. Now, going back to early development, the very first differentiation event takes place with the formation of the blastocyst. You can see here the unorganized cells in the morula gets reorganized to form the blastocyst, the hollow ball of cells shown to the right. Cells in the morula are identical and totipotent. They can give rise to any type of cell found in our bodies. The blastocyst consists of two types of cells, each with different capabilities. Here's a closer view of the blastocyst. The cells on the outside of the blastocyst is called the trophoblast. These cells go on to make the placenta. The cells on the inside are referred to as the inner cell mass. They give rise to the embryo or the baby. These blue inner mass cells are pluripotent. They're capable of producing more than 200 different cell types found in the body. But they're more differentiated or specialized than the cells in early stages of development. Those early cells can make both the placenta and the embryo. This brings us to the first type of stem cells I'll be talking about, embryonic stem cells. Embryonic stem cells used in labs are derived from the inner cell mass. This is why embryonic stem cells are so controversial. They're derived from the cells that have the capability to produce an embryo. I want to talk a little bit more about how embryonic stem cell lines are generated. But before I do that, I have to first briefly mention what happens between fertilization and implantation. Implantation is when the fertilized egg attaches to the wall of the uterus. Fertilization actually happens in the oviduct, the tube that connects the ovary to the uterus. All of the cell divisions I highlighted earlier take place as the embryo travels down from the oviduct into the uterus. Implantation happens once the embryo reaches the blastocyst stage. This is why doctors can now use in vitro fertilization to help with fertility. During an IVF procedure, egg and sperm cells are mixed to allow fertilization to take place. Successful embryos are then cultured for a few days before some of them are transplanted into the mom. In a majority of these cases, the remaining embryos are cryopreserved for later use. But in some cases, around 2.8% of IVF cases in the US, these embryos have to be discarded. Human embryonic stem cells currently in use in the US were derived from such IVF embryos that would have been otherwise discarded. Given their origin, the inner cell mass, embryonic stem cells are pluripotent. They are able to self-renew or continue to grow indefinitely. This is why embryonic stem cells are highly sought after for research involving cell therapy, transplantation therapy, and regenerative medicine. They are also being used for drug development and disease modeling. But there's one major limitation associated with embryonic stem cells. Using them for research purposes, let alone for cell therapy, remains highly controversial. We're now going to go back to early development so I can describe the origins of the remaining stem cell types. When scientists talk about stem cell potential, they're referring to a stem cell's ability to give rise to other types of cells. Remember, the very early cells of the embryo are totipotent. These cells can produce both the placenta and the embryo. The cells of the blastocyst are pluripotent. They can produce either the placenta or the embryo, but not both. Stem cells with greater potential can give rise to more cell types. I'll talk more about stem cell potential in a few minutes. After implantation, the cells of the embryo become more and more specialized. Here's the next developmental stage, which is called the gastrula, when the blastocyst is reorganized into a multi-layered structure. Cells in the gastrula are even more specialized than the cells of the blastocyst. They're organized into 3 germ layers, which go on to produce specific cell types of the body. These cells are multipotent, which means they are capable of producing several different types of cells, but not all types of cells. Here is a more close-up of the gastrula. Cells in the outermost layer, or the ectoderm, can produce skin cells, nerve cells, and pigment cells. Cells in the mesoderm, or the middle layer, produce muscle cells, bone cells, and red blood cells. The endoderm gives rise to pancreatic cells, cells that line our gut, and lungs. Finally, the germ layer is responsible for making either egg or sperm cells. Here is a summary figure of how cell potency changes during development. In the beginning, we have the morula with totipotent cells, where each cell can produce an entire embryo and the placenta. Next, you specialize into the blastocyst. Inner mast cells can produce all 200 types of cells in the human, but not the placenta. These cells are then further specialized when the 3 germ layers are established in the gastrula. Multipotent cells of the germ layers can produce several different, but not all cell and tissue types. At the opposite end, you have unipotent stem cells. These cells differentiate along one type or lineage. The 2 other types of stem cells I'll be discussing, adult stem cells and newborn stem cells, are multipotent. They can give rise to many, but not all different types of cells in the body. All stem cells have the capability to self-renew, or keep producing more stem cells. This is a defining characteristic of stem cells. But I've shown you the potential of stem cells depend on their origin and age. Researchers have discovered adult stem cells in a number of tissues. Adult stem cells are rare and very difficult to identify. What's more, the exact origin of adult stem cells is still a mystery. Researchers can't agree on whether they are leftover embryonic stem cells or whether they arise in some other way. Cord blood and cord tissue are sources of newborn stem cells. The value of these stem cells in research and stem cell therapy is well established. Cord blood contains hematopoietic stem cells. Hematopoietic stem cells were first identified more than 40 years ago, and they are used extensively in stem cell transplants to treat a number of diseases. Hematopoietic stem cells are multipotent, and they give rise to different types of blood cells and immune cells. They're found in both the bone marrow of an adult and in cord blood. But stem cells taken from these 2 sources are not the same. This slide summarizes some of the differences between stem cells from cord blood and the bone marrow. Stem cells from cord blood are at their youngest possible state, which means there's a higher proportion of primitive hematopoietic stem cells in cord blood. Studies show that stem cells from cord blood have higher proliferative potential, 3 to 15 times that of bone marrow. These young stem cells from cord blood have long telomeres, which means they can undergo hematopoiesis, the process of making blood cells, for a longer period of time. When it comes to collection, taking samples from bone marrow involves surgery. Collecting cord blood is non-invasive and painless. The only limitation is that it can only be performed after birth. Before a hematopoietic stem cell transplant is performed, blood samples from the donor and the recipient are compared to see if they're compatible. This is done by looking at markers on the surface of cells called HLA antigens. Studies show that cord blood stem cells allow greater mismatching, meaning that the match between the donor and the recipient doesn't have to be perfect. Researchers believe the immaturity or the primitive nature of stem cells in cord blood is behind this observation. What's more, other studies show that transplants involving cord blood stem cells are associated with reduced rejection and reduced transplant-associated mortality rates. Studies also show that cord blood stem cells have higher repopulation capacity or the ability to replenish the immune system of a patient than stem cells from bone marrow. Given the enhanced potential of cord blood stem cells, preserving them when possible with cord blood and cord tissue banking is recommended to all expectant parents. Let's look at the role of hematopoietic stem cells in a bit more detail. I already mentioned that hematopoietic stem cells give rise to different types of blood cells and immune cells. Given that certain types of blood cells, like our red blood cells, have a very short lifespan, having the ability to replenish them is absolutely essential. Replenishment of blood cells happens largely in the bone marrow. Typically, stem cells produce an intermediate cell type called progenitor cells that are not fully specialized. These cells then produce the more specialized cells that are necessary for function. Hematopoietic stem cells produce two types of progenitor cell types, lymphoid progenitors and myeloid progenitors. Lymphoid progenitors regenerate the cells of the immune system. Myeloid progenitors are responsible for generating several different types of blood cells, including red blood cells. Hematopoietic stem cells are likely the most extensively studied type of stem cells. They're already been used in clinical applications to restore blood cells and immune cells via bone marrow transplants and cord blood transplants. Around 80 different diseases are currently treated with hematopoietic stem cells from both bone marrow and cord blood. Hematopoietic stem cells were explored in detail as part of the Cord Blood Basics seminar series. So if you want to know more, we recommend checking out that webinar. To recap, stem cells are defined by their ability to self-renew and give rise to other types of cells in the body. But not all stem cells are equal. Stem cell potential dictates the number and type of cells they're able to regenerate. Stem cells with higher potency are capable of producing more cell types than a stem cell with lower potency. Finally, I highlighted hematopoietic stem cells as an example of a multipotent stem cell that is currently in use for regenerating cells in the immune system and the blood system. Both adult and newborn stem cells are being explored in clinical trials for their potential for treating a number of highly prevalent diseases like diabetes, heart disease, and Alzheimer's. Still have questions? Email us at info@healthcord.com and we'll be happy to help you.